Today's going to be the last lecture on plants. We're sort of like cruising through all of life, right? So we finished microbes and animals, and today we're going to finish plants. And then on Wednesday, we're going to pick up with the organism that we're sort of most and least familiar with, which is ourselves. And the next 4 lectures are going to be on humans, both looking at aspects of our history, our diversity, and also how genomics is being used to diagnose and treat disease. Okay, so today we're finishing up on plants, and if you remember from last time, we were talking about how you actually identify genes that are contributing to differences in a particular trait. And where we ended up with was this example, which is of a particular trait in rice that is really important for the, domesticated rice varieties. And so if you look at these 2 strains of rice, they're both sort of in a chamber with a fan blowing on them. And you can see that this strain loses its rice kernels very easily. They go flying in all directions, whereas this one doesn't. This is actually a very desirable trait for rice, and that's because you don't want the rice to fall off the plant until you go and harvest it. Okay? And so this is another sort of more general feature of plants under domestication, is that they sort of become a little bit more resistant to the sort of whims of nature, and tougher, so that it basically takes human intervention to get the seeds off of them. Obviously, in nature, this is a desirable trait because an organism is making seeds so that they spread farther and populate a bigger area. So this is an adaptation to nature, and this, we can say, is an adaptation to domestication. So the question then is, well, how do we identify genes that are responsible for a difference like this? Because we might have a strain like Cazalat, which grows in particular environments better than another strain, but we might want it to hold on to its seeds. And so the first step in doing that is to identify the genes that might be responsible for a difference like that. And we talked last time about how you do that by doing crosses and using markers in the genome and finding markers in the genome that are associated with the particular trait you're interested in. So you cross, say, these 2 together, and you get a hybrid plant, and then you cross the hybrid plants together, and you get a variety of different types of plant in that next generation that will have more or less tendency to hold on to their seeds. And then you look at a quantitative measure of that, of how well they hold on to their seeds. And with every marker in the genome, you ask, well, are the ones that are holding on to the seeds more associated with a particular allele at that marker gene or not? And that's how you sort of narrow in on genes that are actually contributing to this difference. So that was done in this example. And the first thing I want to show you is that this is not a simple, what we call Mendelian trait, where it's just one gene that's contributing to this difference. And one indication of that is if you take a bunch of strains of rice and just quantify how tightly they hold on to their seeds, you see that there's quite a bit of range here. So this tendency to lose the seeds is called shattering. And so as non-shattering goes up, those are the ones that are sort of holding on to their seeds tighter. And as it goes down, these are the ones that are sort of, the seeds are much looser. And you can see these are all just different strains of rice that are planted in different places in the world, most of them in Asia. And you can see that the amount of shattering varies widely across these strains. Okay? And when the mapping was done between these 2 strains, where one tends to shatter and the other one doesn't tend to shatter, there were a few genes that were identified as being associated with this particular trait. And in particular, there was one major gene. This is a map of the 12 chromosomes of rice, and everywhere there's a circle is where there was a marker that was associated with the trait. And the marker that showed the biggest association with the trait is in this gene called QSH1. That's just for shattering. Okay? And I don't remember if I defined, I think I defined this term last time. QTL is quantitative trait locus, or quantitative trait gene. That's basically a gene that contributes not just to a Mendelian trait where it's either an off or an on kind of thing, but to a trait where there's quantitative variation within a population. And so we call that a QTL for short. And so QSH1 was a QTL that was identified that has something to do with shattering or not shattering. And this is a gene, and once you have the sort of gene sequence in hand, you can start to do other experiments to figure out why this thing might actually be involved in this trait. And in this example, there's actually a clear experiment you can do that shows a nice clear result about why it might be related to shattering. And that's what's shown on this last slide from last Wednesday's lecture. And it's basically an experiment that's very similar, if you remember, to what we talked about with the stickleback fishes, where you can look at where a gene is expressed, where a gene is activated in an organism by using the complementarity of RNA and DNA and performing some kind of color reaction to show where that RNA is. And so what's shown over here is a real close-up of, you know, one grain of rice growing, and its connection to the plant is through this part here. And what's shown in these two pictures is an even bigger blow-up of just this part of where the kernel meets this stalk. Okay? And what you should be able to see, these figures from this paper aren't great, so I'll point out what you should be able to see, is that, so here in this picture is the stalk part, so that's like this part, and everything from here up is the actual rice kernel part. Okay, and the same thing here and here. Okay, and what you can see here is for this Cazalat strain, the gene is active in this sort of arc that's right at the boundary between the kernel itself and the stalk, and that activity is missing in this Nippon-bare strain. Okay, and if you remember, from this picture, it's Nippon-bare that holds on to its seeds and Cazalat that doesn't. Okay, so there's this extra gene activity in Cazalat, okay, that's associated with losing the seeds that's not there in Nippon-bare. And what we can infer then is that in this sort of ancestral condition, in say the wild ancestor of rice, it's likely that this pattern was there because ancestral rice does shatter. Okay, and somehow the activity of this gene right at this boundary makes it so that in the mature plant the seed is more likely to separate from the stalk. Okay, and the absence of that activity in Nippon-bare and other non-shattering varieties makes it so that there's not as much separation here. Now we don't know exactly what the mechanism is. We don't know why the activity of this gene makes it more separating than the absence of the activity of the gene. But at least this is a first entry into studying that, and it's an important one because it really focuses the attention on these cells here. And the other reason it's important is that this QSH1 gene is not an ordinary gene. Okay, it's a gene that encodes a transcription factor. Okay, and we've seen this before, where important genes in evolution, in dogs, in stickleback fishes, here we go in domesticated rice, encode transcription factors. Transcription factors are proteins that regulate the activity of other genes. Okay, so you can think of them as sort of managers in a sense. They're regulating the activity of other genes, and so it's not just that, you know, expressing one gene here gives you this difference between shattering and not shattering. It's that this activity of this gene probably reflects a large number of genes that are also differently expressed at this boundary that are controlled by the QSH1 gene and activated in Cazalat, but are not activated in Nippon-bare, and so there could be a whole suite of differences between this kind of rice and this kind of rice that make for that phenotypic difference. Okay, any questions about that before we move on to today's lecture? Okay. Okay, so basically what we're doing now is we're just continuing along that same theme of trying to understand where today's crop species came from. And we're going to add an extra wrinkle today, which is a kind of sort of DNA forensics that I think is really, really cool because it allows you to do something that would otherwise be pretty much impossible to do. And that is to read the minds of our ancestors who domesticated these crops. Okay, and so the two questions we're going to address today are, what does genomics tell us about how wild ancestors became today's crop species? So more examples of how particular genes changed, and those were the dramatic changes that caused the difference between this ancestral variety that's not a particularly appetizing thing and this new variety that is. And then how, this is the DNA forensics part, how can DNA sequences tell us if a gene was a target of selection? So what I mean by a target of selection will become more clear throughout the lecture, but basically what I mean is that our ancestors in different parts of the world, which you'll see on the next slide, had these ancestral plants and they turned them into domesticated crops, and changes took place in those crops. And the changes are mediated by differences in genes, okay? And you'll see some examples, which I'm going to show you today, where there's a single letter in the genome changed from, say, an A to a T or a T to a C or whatever, okay? And so today we can look and we can compare the plants and we can do these mapping experiments and figure out what changes occurred. But what we'd really like to know is, were those the changes that, in a sense, our ancestors saw? Were those the changes that allowed our ancestors to sort of select those plants and say, these are the plants that we really want to cultivate from now on, and that's why they became the domesticated variety. So that's a really difficult question to answer, obviously, if all you had at your disposal was sort of archaeology or other non-scientific approaches. You know, there's science in archaeology, but there's not genetic science. One way to think about it is, you know, you can't go back and ask the ancestors what they were looking at. You can't recover the original plants that they were cultivating all the way throughout, because those plants lived and died and returned to the soil. So there's really no evidence, in a sense, that's contemporary with those changes. But what there is the record of DNA changes that's taken place since, and we can use those DNA changes to actually reconstruct what happened at that very early stage when the domestication event happened. Okay, and that's what we're going to talk about today. Okay, so just to give you sort of an overview of domestication, some of the plants on this map will be familiar to you, others you probably will have never heard of, okay? And some of them might be surprising to you in terms of where they were actually domesticated. So we talked already about corn, maize being domesticated in Mexico. We talked about rice being domesticated in East Asia. We talked about wheat being domesticated in the Fertile Crescent. There are a bunch of other species on here that are the earliest domestication events from different parts of the world. So bananas are a very, very old domesticated species, 7,000 years before the present. Squash, beans, also in Mexico. I made this point the other day, but this is really interesting because these three species together basically give you complete protein. Okay, so you can see how sort of major an event it is to complete this trifecta with domesticating the bean in terms of the nutrition of our ancestors in Mexico. The other thing that's really important is that legume species like beans return nitrogen to the soil, so a common farming practice is to grow beans near other plants like corn that require a lot of nitrogen in the soil, and they sort of fertilize each other. Other interesting species have to do with where they were domesticated. Okay, so in many cases, the places we now associate as sort of being most representative of where those species are used or not where they were originally domesticated. So I often think of, say, India and South Asia, Southeast Asia as like the home of the chili pepper. Okay, chilies were actually domesticated in South America. Here. Tomatoes, okay, we think of as sort of, you know, Italy, maybe, home of the tomato. Okay, tomatoes, it's not on this map, but were domesticated in Mexico. Okay, so all of these sort of major sort of culinary changes happened much later in you know, sort of real recorded history with various trade opening up between various parts of the world. In addition to all these plant species, animal species were also domesticated. We talked about sort of the origin of dogs, the origin of cats. Okay, also in the Fertile Crescent, you have goats, sheep, pig, cattle, domesticated for the first time, and these sort of spread all over the world too. The other thing I want to point out is that our knowledge of these domestication events is really sort of continually being updated by new archaeological data and new genetic data. And so from 2006 to 2007, we can even update this map. And there's evidence of squash being found in South America in addition to Mexico very early on, 10,000 years before the present. Okay, now, if you read sort of reviews of this a few years ago, everyone would say, well, wheat was the first domesticated species, okay? But it's actually sort of not clear anymore whether it was, because some of these New World domestication events were quite early as well. Okay, cotton, for example, 6,000 years ago, as another type of species that we obviously don't eat, but was really important in human history. Okay, so let's look at some of these domestication events. We've already talked about corn. You have teosinte, which has this branched appearance, and these things you wouldn't even call cobs, turning into this really tall single stalk with recognizable cobs with kernels on them. We have rice, which we talked about, from a wild ancestor to a domesticated species. Wheat had a very similar change in terms of sort of going from a really fragile state to a really secure state of the seeds. So this is what ancestral wheat or some still existing wheat varieties look like, these sort of tall, slender, fragile seed heads. Most cultivated wheat looks more like this, so really compact, really tough, again, so that none of this falls off before the harvester comes along and gets it. Tomatoes, you'll see more examples later in the lecture of how just dramatic some of these domestication events can be. So that's the ancestor of the tomato, okay? Tiny compared to our modern day tomatoes. Sunflowers and many other flowers are also domesticated, obviously, from what looks like sort of a small yellow unruly wildflower to this sort of, again, really tall plant with huge, huge flowers. Okay? And our knowledge of domestication is really just sort of growing. And so I've included this list of sort of genes that are now associated with changes that took place in some of these domesticated species. And I want to go through this again at the end of lecture when we discuss some of these concepts, but I just want to sort of give you a heads up of the kinds of things we're going to be talking about. So what's shown in this table is a list of genes, okay? For example, here's one involved in shattering, right? So that's SH4. Which crop it refers to, so corn, rice, tomato, wheat. What the function of that gene is in terms of the protein that encodes, and you see this is a theme that's going to come up, is that many of the types of genes that are involved in these domestication events encode transcription factors, transcriptional regulators. And so this column just says that someone has done an experiment to show that this gene actually is directly involved in changing the way the plant fruit looks. And this column is what we're going to talk about some more towards the end, is showing that not only does this gene have something to do with the difference in appearance of the particular trait you're interested in, but also that there's strong evidence from the DNA that indeed this gene, this change in this gene is what our ancestors, in a sense, saw when they were selecting these plants for domestication. And then this column just shows how the gene was mutated to give rise to the version of the gene, the allele of the gene that exists in the domesticated variety. Okay? And so as I said, we're going to talk about this again later at the end of the lecture to summarize. But you can see, just to give you an indication now of sort of, there's a lot of data out there now about genes that are involved in these events from corn, rice, and wheat, but also from tomatoes. You can see a bunch of examples here, that some of which we'll talk about in a second. Peas even, and some more, as time goes on, more and more sort of unusual species too are being investigated in this way. And we talked about some of these genes already. So we talked a lot about SD1, which is sort of the green revolution gene that made these plants short and squat. We talked about just now QSH1, which affects shattering. I want to give you now some more examples from these species that really illustrate how a gene change can actually make a plant very different. Okay? And by inference, you can think about it as making an animal different or making a human different. Okay? The sort of language of genetics doesn't change depending on what species you're talking about. Okay, so let's talk about rice first. First, I need to introduce you to rice, unless you know this already. There are broadly speaking, three main categories of rice. Okay, there's what's called indica rice, japonica rice, and glutinous rice. So indica rice is your typical long grain rice. Okay, what some of us might call white rice. But also sort of basmati varieties and so on are this indica long grain rice. Japonica rice is what we would call sushi rice. Okay, it's a short grain rice. And glutinous rice, sometimes also called sweet or sticky rice, is generally used because it's sweet and sticky. It's another type of short grain rice. The main difference between these is in their starch content. So indica rice is high in a starch called amylose, and japonica rice is high in a starch called amylopectin, and this gives them their different sort of qualities when you bite into them. This is more sort of, I don't know how to describe it, just sort of simple, starchy, and this is more sort of chewy. And glutinous rice is 100% amylopectin, so it's really sticky. Okay? So what are amylose and amylopectin? They're two different ways of storing glucose. Okay? So amylose is just a bunch of glucose molecules strung in a line. Okay? And so glucose, as you know, comes from photosynthesis. And so starch is basically a way for the plant to save the glucose it's produced through photosynthesis. Okay? And if it just takes the glucose molecules, which is a 6-carbon sugar, I'm representing it as these hexagons, and just strings them all in a line, okay, you get the starch called amylose. Okay? Amylopectin is also just a polymer of glucose, so it's just a bunch of glucose molecules stuck together, but the difference is that it's branched. So in addition to just sort of this long linear structure, you have two glucose connecting to the same glucose, and so you get a branch, and so you get this sort of big branched molecule that's amylopectin. And that's basically the difference when you bite into a kernel of rice between it just seeming sort of simple starchy or having a little bit more sort of bite on your tooth. Okay? And there are genes that control how much amylose is made and how much amylopectin is made. And one of those genes is a gene called WAXY. And both amylose and amylopectin are made from glucose by different enzymes. Okay? And these enzymes as a group are called, unsurprisingly, starch synthase enzymes, because they take glucose and they synthesize starch out of them. Okay? So there's a starch synthase that makes amylose, and there's a starch synthase that makes amylopectin. Okay? And specifically, this gene WAXY makes a protein that is a starch synthase that's required to make amylose. Okay? So we can show this schematically here, that if there is a linear chain of glucose molecules and free glucose, and the starch synthase encoded by the WAXY gene is present, then basically the free glucose is added to the end of the existing starch. Okay? And that can go on and on and on and on. Whereas there would be a different starch synthase enzyme that might add it in one of these branch points instead. Okay? So it turns out that indica rice has a normal WAXY gene that produces a functional enzyme. Okay? And that makes sense because indica rice is mostly amylose and very little amylopectin. Okay? And so what I'm showing here is what the gene actually looks like in the genome. So you can imagine a long chromosome extending in both directions. And then the beginning of the gene has an exon, an intron, and then another exon. Okay, so just your standard run-of-the-mill gene. And so as you know, this gene would get transcribed into a pre-messenger RNA, and then that pre-messenger RNA would get spliced to take out the intron, and so the messenger RNA would look like this, exon 1, exon 2, and so on. Okay? And then this would get translated into a protein, and that protein is the starch synthase that contributes to making amylose. Okay, so that's sort of the normal condition. It turns out that in glutinous rice, which does not have any amylose, it has only amylopectin, there's a mutation, a single nucleotide change, okay, that makes this gene produce a non-functional enzyme. And what's interesting about this mutation is that it's not in the coding sequence itself, it's not in the exons, it's not in the promoter of the gene, so it doesn't affect when the gene is activated. Okay? The change is actually right at the junction here at the beginning of the intron. Okay? And we didn't talk much about this, but basically there are punctuation marks written in the DNA letters for where the intron is. Okay? And so if you mutate one of those punctuation marks and change it to something else, then the cell doesn't recognize that as an intron anymore. Okay? And so the messenger RNA still contains the intron. It doesn't splice that out. As a result, when the ribosome comes and starts translating this, it'll start out okay. It'll go through exon 1. It'll make the same protein sequence it would normally make. Okay? But then it runs into a difference here. Instead of going to exon 2, it'll just start trying to translate the intron. As you know, because 3 of the codons in the genetic code table are stops, it's very likely that within a small number of DNA letters here, the ribosome is going to stop. It's going to hit one of those stop codons because the intron isn't meant to code for any protein. Okay? And so that's what actually happens here. So the protein that's made from this mutated gene basically just contains this part and then a tiny little bit, and then it stops. Okay? And that little bit of protein is not enough to produce a functional enzyme. And so glutinous rice versions, glutinous rice strains don't have the enzyme, and because they don't have that enzyme, they can't make amylose. They only make amylopectin, and that's why glutinous rice has 100% amylopectin. Okay? And similar mutations have happened that make the difference between indica rice and japonica rice, but they're not absolute. Because if you completely knock out the function of this gene, you get 100% amylopectin, you get glutinous rice. But there are mutations that can take place in this gene and other genes that sort of shift the balance between amylose and amylopectin, and this leads to different sort of starch content in the different rice varieties. And what's really interesting is this is not just limited to rice. So the same gene, the same waxy gene, exists in corn, okay? And mutations in it have been associated with corn domestication, okay? So what's shown here is basically more of the biochemical pathway that leads to amylose or amylopectin, okay? There are a few steps that happen that are common to both pathways. They happen before this branch point, okay? And there are genes that are known to be different between domesticated corn and teosinte, okay? So teosinte here is written as parviglumis. That's the technical Latin species name of teosinte. So basically all this is showing is that this gene are different between domesticated corn and teosinte, okay? And what's thought to be true is that these changes have made it so that more starch is made, because teosinte doesn't have a very starchy kernel, but modern corn obviously does. And so it's thought that these changes made for sort of greater production of starch out of the starting materials. Okay? And then the changes that are happening here are going to affect the balance of how much amylose and amylopectin you have, and different corn varieties have a different balance of that, so that they're either sort of more sweet or more chewy and so on, better for making cornmeal or for actually eating corn and so on. And so this is the waxy gene of corn, okay, and mutations in it have been associated with a change in this balance. So if you decrease the function of waxy, you get more amylopectin, and if you increase the function of waxy, you get more amylose. And then there are these two other genes. You can also see on this biochemical pathway diagram that at least a few of these have evidence that they were under selection during the domestication process. And again, we'll talk about where that evidence comes from later on. But the point is, not only have these genes changed between teosinte and domesticated corn varieties, but they're likely to be the changes that were actually selected during the domestication process. Okay. What I want to talk about now is sort of the dogs of plant food species, which are different tomato varieties. Okay, so I already showed you the difference between a modern tomato and the ancestor of tomatoes. Okay, but as you must realize from having been to a supermarket at least once, not all tomatoes look like this. Okay, so this diagram is all different tomatoes. There are hundreds of varieties of tomatoes, different seed companies you can get them from. Okay, some of them are very round, some of them are more elongate, some are tiny, little cherry or grape tomatoes, some are these giant beefsteak tomatoes. Some of them are really ugly and sort of have these weird twists and turns in them, but they tend to taste better than the ones that are perfectly round because these were selected to be easy to harvest, and these were just selected because they taste good. And there's usually a trade-off there. It's hard to make a perfect tomato that both tastes good and is easy to harvest and is easy to ship across the country, is easy to sit on a shelf for a while without rotting. And so I'd say it's fair to say that the balance now in our supermarkets is towards tasteless, beautiful, round, unspoiling tomatoes. But if you go to the green market during the summer and you pick up some of these, you'll say, oh, so that's what a tomato tastes like. So enormous amount of diversity within tomatoes. The other thing I should point out is that you might not realize what species are the close relatives of tomatoes because, again, the differences between them are just so gigantic. So does anyone know the two species that are basically the most close relatives of tomatoes that we also eat? Any guesses? Pepper is close. Okay, so they're more like a distant cousin. Nope. So closer than peppers. There are two species that you all eat. Well, one of them you eat tons of. The other one you probably eat also, that are close relatives of tomatoes. Anyone know? Nope. So cucumber is more related to squash. So I'll give you one of them, which is not as common, but might be easier to recognize as close to a tomato, which is an eggplant. Okay, so tomatoes and eggplants are very, very closely related species. Obviously very different looking too. The other one that's a very close relative of tomatoes, you probably would not guess in a really, really long time, and I'm not going to wait, is potatoes. Okay, so potatoes, eggplants, and tomatoes are very, very closely related species. And you can tell just by the difference between a potato plant, where we eat actually what's in the ground, and a tomato plant where we eat the fruit, how different some of these plant varieties can be from one another. So if you go a little bit farther out, then the relatives of tomatoes are also pretty surprising. So peppers are relatives of tomatoes, but another close relative of tomatoes is tobacco. Okay, so all of these species are so-called nightshade species, so they typically contain sort of poisonous or sometimes hallucinogenic compounds. Okay, which is what makes tomatoes actually easier to grow because animals don't eat the plants because they're poisonous. Another relative of tomatoes for Harry Potter fans is mandrakes. Okay, so these like really nasty root things that look sort of like really ugly humans. Okay, and that's why they sort of got mythologized as sort of having these supernatural human-like qualities, and also because they're poisonous. They make you hallucinate, basically. So a lot of diversity within very, very closely related organisms. And what that diversity allows you to do is then try and understand how genetic changes actually create that diversity. So one simple example of a kind of trait that's different between different varieties of tomatoes is just whether they're round or pear-shaped. Okay? So you've probably seen a lot of tomatoes like that and a few tomatoes like this. And it's now known the major genetic difference that leads to a tomato appearing like that or like that. And again, this is just one nucleotide difference, okay? Within the enormous genome of a tomato plant, one change from a G to a T gives you a shape like that going to a shape like that. Okay, and this example was figured out a few years ago. And basically, it's in a gene called ovate. And this is the way the gene looks like in a normal round tomato variety. Okay, again, exon 1, intron exon 2. And then the sequence of the protein, the coding sequence, it starts with ATG, obviously, and goes on and on. And in this part of the protein, there's a GAA codon. GAA, if you look it up in the genetic code table, codes for glutamic acid. And then later on in this exon is TAG. So UAG, if you look it up in the genetic code table, is a stop codon. Okay, so there's the coding sequence of this particular gene. And the difference between a round one and a pear-shaped one is that this codon, GAA, in the pear-shaped varieties, it reads TAA. Okay, just a single nucleotide. But TAA, if you look up UAA in a genetic code table, is another stop codon. Okay, so again, what you get here is an incomplete protein. So the protein ends here instead of translate, 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 and then stop. Okay, and just that difference, that making this protein non-functional, makes the fruit round. Ovate, pear-shaped instead of round. Okay, so that's all well and good. I can tell you that this genetic difference makes for this difference, but how do you actually show it? So if you were the people, if you were in the lab that sequenced these genes and found that this one had a G and this one had a T, and you said, aha, I think I found it. I think I found the gene that makes for this difference. How would you go about actually demonstrating that this change is what makes for that difference? How do you do that? You've already seen a couple examples of how you do this kind of experiment. What would you do?
Does it have something to do with like what we saw in last lecture, where you have to grow a number of the plants and then determine where on the chromosome how many plants each stage?
Right, so that's what led to the identification of this gene as the candidate gene that causes this difference. Right, so that's what gets you there. But then once you get to this gene and you say, okay, there is this difference, I need to demonstrate that this really is the gene and it wasn't just an accident that I found this difference. How do you do that experiment? How do you show that this change is what creates this change? Any guesses? What you do is you take a variety like this and you introduce this gene into it. Okay, because what I told you is this looks like a non-functional gene. Okay, it has a premature stop codon. Right? This looks like a functional gene. Okay, so if this really is the difference, then I should be able to take this gene, put it into this variety, and get a round tomato out of it. Okay, that would show that this difference is the causative one for that difference. Okay, and so that experiment has been done. So we start with a normal round variety, which has a functional ovate gene, okay, and a normal pear variety, which has this mutated ovate gene, okay. And then this is what happened if you take the pear variety and put in the ovate gene from the round variety, okay. That's the only part of this genome that you're adding to this genome, okay, and lo and behold, you get a nice round tomato. Okay, so that's proof. That shows that this is the gene that causes this difference, because you can repair this difference by just putting that gene back in. Right. Right. So this gene did not convert this into that. Right? It converted this into a small yellow round tomato. Okay. So there must be other genes that control the color difference, right, and control the size, right? And you put all of those together, and you can go from this to this. So in principle, if you identified by the same types of experiments the genes that would make this red or make this bigger, and you can combine those genes with this gene all in this, then you might be able to actually recreate something that looks exactly like that. Okay? And some of the genes that are involved in the size and shape of the fruits have actually been identified too. So here's one example. Okay? There's a gene called SUN, and there's a nice correlation between how active the gene is and how long the fruit is. Okay? So on this axis is the amount of mRNA produced by this SUN gene. Okay? So how active the gene is, how much transcription there is of that gene. And on this axis is a measure of how much it looks round and how much it looks long. Okay? So rounder ones down here, longer ones up here. And what you can see is there's this correlation. So wherever there's a low amount of SUN messenger RNA, so a low amount of gene activity, those tomatoes tend to be round. Okay? And wherever there's a higher amount of SUN messenger RNA, so higher activity from this gene, they tend to be long. Okay? And you saw a correlation like this before, where instead of looking at how much messenger RNA there was, we were looking at how many repeats of a repeated sequence there were in a dog gene, or in Huntington's gene, or in the Flo11 gene of yeast. Okay? So we're associating some aspect of the genetics, so in this case, how much activity of the gene, to the actual phenotype, the shape of the fruit. Okay? And it's a pretty good correlation. Okay? But then there's these 2 points down here. So what are these 2 points down here? What shape tomatoes do these correspond to? They're round. Okay? They're down here. Okay? How much activity of the SUN gene is there? There's high activity. Okay? So these two are sort of an anomaly. They don't fit the correlation. So what do you think is going on with these two? Mutated for where would the mutations be? What would be mutated? So a gene would have to be mutated, right? Not the phenotype. Okay, so which gene would be mutated? The SUN gene, and how would it be mutated? Okay, so one hypothesis, you're sort of like 99% of the way there. One hypothesis would be that even though there's a lot of messenger RNA from this gene, the protein it makes is non-functional. Right? So something like that. So you'd be making a lot of the messenger RNA, but it would do you no good because you would still have basically no activity of the protein. And low activity of the protein is associated with round fruits. So that would be a hypothesis that could maybe be tested by looking at these two particular strains. But first I want to show you that this inference based on this correlation is actually true. So this general connection between the amount of SUN mRNA and the fruit shape is definitely true because you can do an experiment. So you can take a normal round variety, okay, and you can engineer it. And remember, that's usually associated with low activity of this gene. And you can engineer it to have more activity, okay? And we'll talk in later lectures about how you actually do this engineering. But when you do that, you convert this into something that makes these long tomatoes, okay? And you can do the opposite. You can take a normal long variety, okay, the sort of oval shape, and engineer it to produce less SUN messenger RNA, and then you get more round fruit. Okay, so that connection is real, and that's the same type of experiment, again, that you use to sort of definitively show that a particular gene is involved in a particular trait difference. Okay. One more final example of these kind of trait differences is back to teosinte and corn. And as you recall, there are lots of differences between teosinte and domesticated corn, and I just want to play you a little clip from this video I played parts of this video before when we were looking at the stickleback fishes, because it gives you sort of a nice visual on the difference between corn and teosinte.
So although now widely planted around,
obviously there are scientists who are interested in figuring out what changed between teosinte and modern domesticated corn. And so one of the clues is this mutation that you can get in some corn plants that makes them look like teosinte. Okay? So this is a corn plant, normal domesticated corn, except it has this gene that's been mutated. And so instead of one long stalk, it has multiple stalks. Okay? It has this more branched architecture like a teosinte plant. The gene was therefore called teosinte branched 1. Okay? And it turns out that this gene actually, not only is it just, you can get a mutation in a modern variety and get something to look like teosinte, but it actually was this gene that changed from the ancestral teosinte species to domesticated corn. And we're going to use this as our example for how you then show that it was actually humans thousands of years ago picking corn plants that had changed because of a mutation in this gene. Okay? And so first, a little bit more about this gene. Basically, the difference between teosinte and corn in this gene is one of, again, activity, how much mRNA it's producing. Okay? So the teosinte version of the gene produces a little bit of messenger RNA. It's not very active at all. Okay? And so I'm schematizing this here by showing the coding sequence of the gene that would have the amino acid codons in it, and then the promoter of the gene, which determines how active the gene is. Okay? And this protein, when it's translated, it suppresses the formation of lateral branches. Okay? But because its activity is so low, teosinte plants actually have branches. They have multiple stalks growing up from the ground. Okay? The modern corn version of the gene is much more active. Okay, so there's been some change in the promoter that makes it a more actively transcribed gene, so you get much more messenger RNA out. Because you get much more messenger RNA out, you get more protein. And because the job of the protein is to suppress the formation of these branches, then corn grows without any branches. It's just one stalk. Okay? And so we now know that this is a major difference between teosinte and corn. And the question is, how do we show then that not just we can see this difference, but our ancestors who were domesticating corn were actually seeing this difference? Okay? That's not a question you can get at through archaeology or history because it happened too far ago in the past. So we'll get back to that, but first I want to show you schematically how you do this kind of thing, how you do this sort of forensic analysis to figure out which gene was actually the target of selection. Okay? And so I'm going back to my favorite schematic plant example, which is these yellow and red flowers. Okay? So imagine that you're sort of living thousands of years ago, and you have this field growing nearby of yellow flowers, and one day you're walking around in your field and you see a red flower, and you say, wow, is that beautiful or what? I really want more red flowers. Okay? So what I'm going to do is I'm going to not really pay too much attention to the yellow flowers, but if I see this red flower, I'm going to guard it carefully, and when it produces seeds, I'm going to make sure I plant all of those seeds, and so that next season there are going to be more red flowers. Okay? So you do that, right, and in the next generation, there are proportionally more red flowers than there were before. Okay? And then you do the exact same thing. You have this little plot where you have your red flowers, you guard them carefully, you make sure no animals eat the plants, and you make sure you get every single seed out of them, and you grow them, and you plant them again, and you guard that territory again, and so in the next season, you have even more red flowers, okay? And then finally, if you do that enough, you get sort of this pure breeding variety of red flowers, okay, from this original single plant that produced red flowers, okay? So that's sort of, in a very simplified version, what domestication is. Okay, it's choosing something that has some desirable property and intentionally growing it to the point where that's all you have, and you don't have the original ancestral version anymore. Okay, so now let's look at what happens at the genetic level while you're doing this. Okay, so what I'm going to do here is imagine that there's one gene. Okay, and this generalizes to when you have more than one gene controlling a trait. But let's just, for simplicity's sake, say there's one gene that causes this difference. And so there's an allele of the gene that's associated with the red flower color and an allele of the gene that's associated with the yellow flower color. Okay? But these yellow flowers have been growing for a really long time out on this plot of land you have. Okay? And so as time goes on, they accumulate mutations. And each plant will tend to have some mutations that are different from other plants. Okay? And the reason is that the mutation is sort of this inexorable process. It just happens, and as time goes on, you accumulate more and more mutations. That's why, between two humans, 1 out of every 1,000 nucleotides in our genome is different, okay? And most of them have no effect on sort of our appearance or our physiology or anything. They're just differences, okay? So we can label these yellow alleles actually as different alleles, okay? They all produce yellow flowers, but each one has been around long enough that it has different mutations than different other ones, and so we can label them yellow 1, yellow 2, yellow 3, and so on up to yellow 14. Okay? And then I'm going to say that this one that has red flowers has an allele of that gene that I'm going to call yellow star. It has a mutation that not only makes the sequence different from these other yellow alleles, but actually makes it produce red flowers. Okay, I could have called that R or something, but I'm going to call it yellow star. So the star means like that key mutation that happened. Everyone with me so far? Okay, so that's our starting point. And then what happens in the next season is that these yellow plants are parented by yellow plants in the previous generation, but all of these red plants came from that red plant. Okay, they basically have one parent for all of these plants. Okay, and you could say, well, okay, so maybe this plant gave rise to this plant, and so they have the same yellow allele, yellow number 5. And this plant gave rise to this plant, so they have the same yellow allele, yellow number 9, and so on and so on and so on. Okay? But all of these red ones have this yellow star allele. Okay? Next generation, same thing happens. Okay? Let's say this plant gives rise to this plant, so they have the same yellow 9 allele. This plant gives rise to this plant, yellow 4, but maybe a mutation happened. Maybe another letter of the DNA code for this gene changed so that instead of yellow 4, you have what I'm calling here yellow 4 star. Okay, it's still a yellow allele. It still produces yellow flowers, but it has a change that distinguishes it from that one. Okay. This one gives rise to this one, and so on. And then all of the red ones are yellow star alleles that produce the red flowers, okay? But again, there could have been another mutation that doesn't change the color of the flower, but makes this allele distinct from these other ones because, say, this parent gave rise to this one, and in the process of making the pollen or the egg, it copied the DNA a little bit wrong, so there's one letter difference here, okay? And so these aren't all identical anymore, but they're very, very similar, maybe with 1 or 2 changes, okay? And then in this final generation, when sort of the domestication is complete, we have all yellow star alleles, and some of them are yellow star star, okay, because they came from that plant instead of these other plants. So this is the end result. You have mostly yellow stars and then a couple of yellow star stars, okay? So now let's look at it and see if that makes any predictions about what domestication does to genetic diversity. Okay? If you look up here, basically what I said was each plant, even though most of them are, almost all of them are producing yellow flowers, they have different alleles. They have what we would call a high genetic diversity. And that diversity doesn't necessarily translate into phenotypic diversity. It's just genetic diversity. Their DNA is slightly different. They have A's, some of them have A's and some of them have T's in a particular place, but it doesn't change the color of the flowers. Okay? But down at the bottom here, we have something very different. On the surface, it's not that different. Up there, we had all yellow flowers, and down here, we have all red flowers. But the genetic diversity has changed quite a bit. We went from having a lot of genetic diversity at this gene to having next to none, okay? Because all of these plants share a common ancestor in the very recent past, okay? Only, say, 3 generations back. And so all of these yellow alleles, yellow star alleles, are identical, okay? And then there are a couple with maybe one change from that. This is a really, really, it might not seem like it yet, but this is a really, really powerful concept, okay? Because what it allows you to do is just collect DNA samples from a species for any particular gene and ask how much genetic diversity is there among those DNA sequences compared to other genes in that same species. The ones that have low diversity, we infer, went through a process like this, where they were selected, where they contributed to a trait that really mattered, and that was selected through this kind of process. Now, this is what we would call artificial selection, in the sense that some human being intentionally chose a particular plant to make it overrepresented down here. But the same is true of natural selection, okay? And I'll show you an example at the very end from humans, where the same kind of data, basically on how much genetic diversity there is at a particular gene compared to other genes, tells you that the change that occurred actually was subject to selection, that it was giving an advantageous phenotype that persisted, whereas the previous phenotype went away. Okay? So how does this actually work with an example like this teosinte branched 1 gene, which we know has something to do with how the teosinte plants versus the corn plants look? So remember what I said, which is that the important change in this teosinte branched 1 gene didn't happen in the coding sequence of the gene. It happened in the promoter region, because the difference between teosinte TB1 gene and corn TB1 gene is in how much messenger RNA it makes. Okay? So the corn gene produces a lot more messenger RNA than the teosinte gene, and the change is a nucleotide substitution somewhere in this promoter region, not in the coding region. So what we can do is we can get a bunch of corn plants and a bunch of teosinte plants, get the DNA sequence for this whole region, and look at the genetic diversity in the promoter region and in the coding sequence region. So in a sense, the coding sequence region is our control for how much genetic diversity there is in the promoter region. If this gene was the subject of selection, if this gene, this change in this gene was actually what human ancestors in Mexico thousands of years ago saw, then the genetic diversity in the promoter region should be very low, and the coding region should be higher. And that's how we know, because the coding region diversity would be higher than the promoter region. So let's look at some sequences, because these are not the actual sequences, because the actual sequences are really long and there are lots of them, so this is just sort of a play version of it, but it basically has the same properties as the actual sequences. So what I'm showing you is the sequences from 3 corn plants, so modern domesticated corn, and 3 teosinte plants, and a bit of sequence of the same part of the gene from the promoter region and from the coding sequence region. Okay? And so first let's look at the coding sequence. What you can see is most of the positions are the same. That's because, you know, this is the same gene. Okay? It's encoding the same protein. But there are a few positions that are different. Okay? G or C in this position, A or C in this position, A or T in this position. So there's a good amount of genetic diversity then. There are 3 out of whatever, 10 positions that have differences. Okay? And what's important also is that the diversity is not partitioned between maize and teosinte. So you can see that maize has either G or C here, and teosinte has either G or C here. Maize has either A or C here, and teosinte has either A or C here. Maize has A or T here, and teosinte has A or T here. So just by looking at these sequences, you can't tell if a plant is teosinte or if it's maize. Okay, and there's differences. There's differences among the sequences, but they don't partition according to maize or teosinte. Now, if we look at the promoter region, what do we see? We see the same kind of diversity in the teosinte alleles. So at this position, this position, this position, this position, this position, we see different nucleotides in different sequences. Okay, so that looks similar to what we see here. But that's limited to just the teosinte plants, because if you look at the maize plants, these sequences are almost identical. Okay? In fact, there's only one difference in this plant with the other plants. Otherwise, these sequences are exactly the same. Okay? So this is exactly the pattern you would see if what happened was sometime in the recent past, someone picked out a teosinte plant that looked a little funny because it didn't have as many branches and started planting that more. Okay? And when the seeds came from that, planted those. Okay? Because what that does is it creates what we call a genetic bottleneck. It takes one plant to sort of repopulate that whole variety. Okay? And by doing that, you get one DNA sequence instead of a diversity of DNA sequences. Okay? So this was exactly the evidence that was gathered, okay, to show that this gene not only causes this phenotypic difference, but, and this is remarkable, is the gene that our ancestors saw. The effects of this gene is what our ancestors saw. Okay? And so we can summarize this way. If we look at genetic variation, it's low in the promoter region and high in the coding region. Okay? For some other gene, it might be reversed. If the important change in the gene was in the coding region, then we'd expect the pattern to be reversed. High variation here, low variation here. Okay? If we look at the corn alleles and compare them to one another, then what we see is that the corn alleles for the coding region are indistinguishable from the teosinte alleles. Okay? It's sort of, you can't tell which is which just by looking at that sequence. Okay? But the corn alleles in the promoter region are all clearly related to one another through very recent common ancestry. Okay? And that's the tip-off that some selection event happened, where a small proportion of the population was chosen as the ones to sort of keep, and the rest were discarded. Okay? And so from this and this, we conclude that this region was affected by selection and this region wasn't. That is, this is, by looking at the corn plants, this nucleotide change in this region is what our ancestors saw when they domesticated corn. Okay? And so it's evidence like that that lets us fill in this column of the table. Okay? So if you remember the structure of this table, we have a gene, and so the first gene on this table is in fact TB1 from corn. Okay? Now the function of this gene is it's a transcriptional regulator. So again, this is a theme, right? This is a transcription factor, a protein that controls the activity of other genes that's changed. Experiments have been done to show that this gene actually does cause changes in the phenotype of the kind I showed you for the ovate gene. What I just showed you is that there's evidence from the DNA sequences that this was a target of selection, that the domestication process did, in fact, involve this gene and this particular change in the gene. And that change was a regulatory change. It was a change in the promoter region of the gene. It wasn't a change in the coding sequence of the gene. Okay? And so we can go through the rest of the table and basically see these things popping up again. So TGA1 is another gene that was involved in the domestication of corn. Again, it's something that encodes a transcriptional regulator, but it doesn't have anything to do with how the branching of the plant happens. It actually has to do with how the seeds are encased. Okay? And as you know, the teosinte cob is very different looking from a corn cob, and so a bunch of changes happen there. And this is one of the genes that contributes to that difference. Experiments have been done to show that that gene actually is the one that causes that difference in the seed casing. And experiments have been done to show that that gene was under selection. Okay? In this case, it wasn't a change in the promoter of the gene. It's actually an amino acid change in the protein itself. Okay? So in this case, the evidence would be sort of reversed. The promoter of the gene would be expected to have high genetic diversity, and the coding sequence of the gene would be expected to have low genetic diversity. Here's another one we've talked about, right? QSH1 in rice, transcriptional regulator. It is expressed on what's called that abscission layer, and it affects the shattering of the rice. It's been shown to directly affect the phenotype. It hasn't been tested yet. whether the DNA evidence of the different rice varieties shows that that was something our ancestors saw when they were domesticating rice. We don't know that yet. This one, again, was a regulatory change, which you saw. The coding sequence of the gene didn't change, but where it was activated changed. Okay, whether it was activated in that abscission layer or not. Okay, a bunch of other genes. Again, transcriptional regulators pop up again and again. Here you have rice, the color of the seeds. Another gene that has to do with shattering. In tomatoes, you have one that has to do with how heavy the fruit become at maturity. Here you have one in wheat. Inflorescence structure is just what the flowers look like, basically. So again, a trait that's important between domesticated varieties and their ancestral varieties. All of these have been shown to be the ones that really cause this difference, but most of them have not yet been tested to see whether they were actually the target of selection. The list goes on, and we're not going to go through every single example here, but the main themes, again, are transcriptional regulators are all over this list. And then the other major category that's on this list is enzymes. Okay? Isoamylase has to do with starch production, and you can see here's a gene where it encodes an enzyme that is involved in starch production, and the phenotype it affects is whether the corn is sweet or not. Okay? Another enzyme, okay, another transcriptional regulator, right? And in some cases, these have actually been shown to be changes that were selected under domestication. Again, so even in peas, okay, which aren't studied very much, here's another enzyme that has to do with starch production and sugar content in the peas. A gene has been identified that controls that phenotype. Okay, enzyme, transcriptional regulator, enzyme, transcriptional regulator, transcriptional regulator, transcriptional regulator. And so I've shown you examples in the course already, as I said, in dogs and sticklebacks and so on, where transcriptional regulators were important. And in humans, you might imagine that the traits that are specific to sort of what makes us us, I would say a very strong prediction is that changes in transcriptional regulators would be involved there too. You might be a little skeptical that changes in enzymes might have something to do with humans, okay? You know, because all of these enzyme examples have to do with how much starch you make of different types. Okay, so I want to conclude with two examples of two enzymes that are different in humans from our ancestors that have a really important effect on our phenotypes, okay? One of them has to do with circadian rhythms, which is our sort of daily rhythms, the fact that we don't like to get up earlier than we normally get up, the fact that if it's late we start to get really sleepy, the fact that when we travel long distances we get jet lag because our internal body clock doesn't match the external time. Okay? So here's a gene and a mutation in that gene that causes a rare sleep disorder. Okay? And the sleep disorder is one where the people who have it have what's called a fast biological clock. So basically what happens is instead of having a 24-hour clock, they might have a 23-hour or a 22-hour clock. So they go to sleep at a normal time at night, and they get up too early. Okay, and if they keep doing that, they just keep getting up earlier and earlier because their internal body clock is telling them, well, it's time to get up. But it's only, 22 hours later than the previous day instead of 24 hours later than the previous day. Okay? As you might imagine, that can be a very, very debilitating condition to have, right? Because as you probably know, when your body's telling you to go to sleep, you really need to go to sleep. The gene that's involved here is actually an enzyme. Okay, so we have enzymes that are involved in setting our biological rhythms, and these can change. And when certain changes happen, they cause disease. They cause a disorder where you don't get up at the right time. But they probably also contribute to just sort of natural variation among humans and how sort of tied we are to our biological clocks, how easy it is for us to adjust to jet lag and things like that. The other example is a really, really important example having to do with lactose intolerance. Okay? And so Most of you are probably aware of what lactose intolerance is. How many of you have lactose intolerance? Yeah, it's, you can see what the percentages are here, okay? So 75% of African Americans, 90% of Asian Americans have lactose intolerance to some extent, okay? And basically what it means is when you're an adult, you can't digest lactose, so you have trouble eating milk, drinking milk, and you get all, you know, stomach ache and sometimes very, very uncomfortable. And what I want to point out is that actually that is the normal condition of humans, and the rest of us who are lactose tolerant are actually the mutants, okay? Now why is that? The reason is that we're all mammals, okay? And as mammals, when do we drink milk? We drink milk when we're infants, okay? And our mothers are giving us milk, right? So when we're older mammals don't drink milk as a rule, okay? And humans only started drinking milk about 10,000 years ago when we started domesticating cattle, okay? And realizing that the milk from cattle could actually be a nutritious source for our diet. But as adult humans at that point in time, most adult humans were unable to digest the milk properly, okay? And so there was a lot of stomach aches, there's a lot of other things going on. But one lucky human got a mutation that made it so that their lactase gene, the enzyme that converts lactose, and allows you to digest it, so that enzyme was actually inappropriately, in a sense, activated late in life. Okay, so not only as an infant can you digest milk, but as an adult you can digest milk. That was an advantageous change because by co-occurring with dairy farming, it allowed people to get a lot more nutrition than they otherwise would. Okay, and so us mutants, okay, have a different version of the lactase gene that's not only different from the people who are lactose intolerant, who we can say are normal, okay, but different from every other mammal. Now, the story I just told you is one where, you know, the nutritional value of it made it advantageous to have this change, okay? And we can actually look to the DNA evidence to see if that was actually as a target of selection, if our lactase gene was a target of selection, because if it was, then you expect a low genetic diversity of the lactase gene among the people who can tolerate lactose.